I cannot see it on the computer.
Good morning, everyone. My name is Maya Luria with TMC for Seniors. And today I am thrilled to have Daniela Zarnescu here with us. She is a PhD. She is joining us from the University of Arizona, and she's with the College of Science. She has been researching and teaching for over 15 years, and she earned her PhD in biochemistry and molecular biology at Penn State University. Dr. Zarnescu is a professor in several fields, including neurosciences, genetics, molecular and cellular biology, and her research focuses on the molecular mechanisms of aging and disease-related brain degeneration. So today she's going to be discussing stress resilience and how that can lead to a longer life and a stronger brain. So I want to welcome you here today, Dr. Zarnescu. It's wonderful to have you, and we're very excited. This is all part of our Brain Health Month. And so I know you're going to be able to share quite a bit with us. I'm going to go ahead and put your presentation on the screen for you. And you are welcome to join us. Also, I'm sorry, before you get started, I do want to say that we are also on live. We are on live on our YouTube channel as well as our two Facebook pages. And in addition to that, we have an in-person audience. So bear with us. We are just getting everything together so that we can present to you in a hybrid manner. So welcome.
Thank you so much, Maya. So not everyone in the room heard your very nice introduction. Thank you for that. So I actually wanted to introduce myself to you anyway. So what Maya did is she just gave a short biography of my career trajectory. So I actually wanted to start that way because I started off as a physicist. I have a bachelor in physics from University of Bucharest in Romania, and then I had an opportunity to continue my training in the United States, and I became interested in molecular biology and genetics. And so I got my PhD in biochemistry and molecular biology at Penn State University. And then I continued my training. It's quite a long training, actually. This academic track involves a PhD and then a postdoctoral training stage. And I did that at Emory University Medical School in Atlanta, where I became interested in using simple model organisms like fruit flies to study the molecular mechanisms of human disease. And I'll tell you more about that if you're wondering how can that be. So I joined the University of Arizona in January 2006. And I'm a professor now in molecular and cellular biology, but I have joined appointments in neuroscience and neurology, and as Maya said, in genetics. I'm also a member of Bio5. So I consider myself a truly interdisciplinary researcher. If someone asks me, what are you? I'm not quite sure if I should say a geneticist or a neuroscientist. I am a curious scientist who investigates the molecular mechanisms of neurodegenerative disease. And in my lab, as we've made progress in studying neurodegeneration, we realized that we cannot ignore aging. Aging is the biggest risk factor for neurodegenerative disorders, as well as cancer and other diseases. And we cannot understand neurodegenerative disease if we do not study and understand the normal process of aging and what might go awry in normal aging that can cause neurodegenerative disease. So that's kind of how I got into the field of aging. It's more of a recent interest. So what I'll tell you today is about how we became interested in stress resilience at the molecular level, how we can use those simple little annoying little fruit flies that you may be annoyed of in your kitchen to really understand basic molecular mechanisms that give us insights into human biology and mechanisms that underlie human disease. And then at the end, I'll show you some unpublished data, so you cannot tell that to anyone when you leave this room or else. It's a new and very exciting project that we started with funding from the Innovation in Aging Initiative at University of Arizona. So I'm super excited to share that with you, but it is very preliminary, and I will tell you where we want to go with that. So please feel free to interrupt and ask questions. I think that will make it for a much more fun and interactive session. So, oh, I'm supposed to use this. Maybe I can use this. This worked when I tested it.
Try now.
Ah, all right. So if you, I put this talk together at the last moment because this is a really busy semester for me. I teach 3 days a week. I do study section. I went to meetings. It's been a little busy. So as I was preparing for this talk on stress resilience, I did a Google search. If you type in stress resilience, you're going to get thousands and thousands of hits, lots of overwhelming information. However, the essence of it all is as defined by the American Psychological Association, that basically defines stress resilience as the process of adapting well in the face of adversity, trauma, tragedy, or threats or significant sources of stress, whether they're family or relationship problems, serious health problems, workplace or financial stress. They might as well add COVID or pandemic to that, right? I mean, we've just coming off a very, very stressful period in all of our lives. So that would be the essence of it. And the reality is that we cannot escape stress, right? We all encountered or were exposed to stressful situations as we drove to be here today. There's stress in our lives at every moment and in any circumstance. Yet some people deal better with stress than others. I'm sure you all can recognize that. So when I started going to the meetings held by experts in aging and clinicians at University of Arizona that try to, gather many of us to be interested in aging and work on aging, a lot of what I've heard is, we're interested in stress resilience. Why is it that some people age better, are less stressed, live longer? And it's not just about living longer, it's about living longer and healthier. My mother is 85 years old, and she still walks around, but she cannot go by herself outside anymore. But she's still, reasonably healthy. So what we want is to live as long as possible, as healthy as possible. Not just live longer, but also be healthier. So when I was hearing this, I thought, okay, clearly that's an area of interest. But I was asking them, well, I'm a molecular geneticist, right? I'm interested in molecules and mechanisms. I do care about people, but I really want to know what's happening in the cells, what's happening with molecules. What does that mean at the molecular level? And no one could answer that question. So I realized that here's an open field of investigation. We have no idea what stress resilience means at the cellular and molecular level. So last night I was that person. Now I'm more that person to the right because I kind of put together a reasonable, what I hope it's a logical train of thought with what I want to share with you today. So one of the things that probably wasn't in the textbooks when you went to school is that actually cells do know when stress is around them. Cells actually have very well put in place evolutionarily conserved mechanisms to respond to stress. And that stress can be, that stress can be starvation of nutrients, could be problems with proteins not forming correctly and clumping and forming aggregates, could be problems with viral infection. Yes, viruses cause stress granules to form, oxidative stress, changes in pH. So when all of those things happen to cells, cells actually respond by forming, I hope you can see this puncta over here, these little granules that we call stress granules. So just like the name says, when cells are experiencing stress, they form stress granules. And you don't need to worry about all those molecules and arrows, that's for me to worry about. But essentially, there are molecules, there are proteins and RNA inside cells that when the cell senses stress, they say, we're going to come together and we're going to form these granules. And the idea is that these granules form to allow the cell to deal with the stress. And these granules sequester important proteins and important RNAs and let the cell deal with the damage or the insult that it's exposed to. So I have some movies to show you how cells actually respond to stress. Would it be possible to turn some of the lights off? Or I don't want to put you to sleep, but that would be a test if I put you to sleep or not with my talk. Okay, so I think you can see that a little better. We can visualize these stress granules as they form. We can figure out what's in them, and we can look at them as they form in response to various stresses that the cells are exposed to. So what I want you to look at next is those cells labeled in red. The red corresponds to a protein. It happens to be an RNA binding protein that is kind of the seed for these stress granules when they form. And these cells are currently exposed to just media, just a good, nice, nutritious media. And then at some point, the investigator is going to add arsenite, which is a form of inducing oxidative stress. So as I start this movie, let's take a look at the red cells, and I hope you see how from that kind of uniform sea of red protein, there's still no drug. At some point, you're going to see there at the top left that the investigators are adding drug right now. Don't worry about those little blobs. Those are cells dying. And so I hope you see that that sea of uniform red becomes granular, and it's quite dynamic. Those are stress granules or RNA stress granules that form when the cell is exposed to oxidative stress. That happens to cells in our body on a daily basis, okay? But cells are able to respond to that. Sorbitol is a major component of chewing gum. At high doses can cause stress, and those kind of granules form. Changes in pH, even mechanical injury. These granules were initially identified in immune cells. So they are present in every cell type in the body. Of course, I'm interested in the nervous system because that's the coolest part of our body. But they form in heart cells, in muscle cells, in blood cells, everywhere. And every cells respond differently and in different ways in response to stress. What's common is that these granules form initially as a protective measure. So let's look a little bit more in depth into that. Here on the left, you have now the protein is labeled in green. You're going to see how upon, I think it's also oxidative stress applied here, the protein that's labeled with the green fluorescent protein, initially kind of uniformly, nicely localized in the cell, will form granules. Let's take a look at that. And you can see that in this particular case, in a matter of minutes, you start seeing these granules forming. And after 30 minutes, they are pretty big, they're moving around. The cell knows it's been exposed to oxidative stress and it's forming these. So what we now know about the stress response is that it initially forms as a protective measure. Cells are forming these and are saying, we're going to stay put and keep a low profile while the cell is trying to deal with the damage. And if the stress lasts just a short, reasonable amount of time, which can vary depending on the cell type, then the stress granules can dissipate and everything goes back to normal. And it's like, we're good again. This is called homeostasis. So if the stress is just short and not too profound, those stress granules, if you wash the stress away, I hope you can see how they start diluting out a little bit. And by the end of the experiment, you see fewer and fewer of these puncta and more diffuse protein. So in this particular case, within an hour and a half, this cell was able to deal almost perfectly with the stress that it was exposed to, and it dissolved or dissipated the stress granules, and it restored to health or homeostasis. Yeah.
Do these granules hit all of your cells at the same rate and force?
Excellent question. Cells have their specificity, so different cell types will respond differently in a different time manner and in different ways. The granules may be smaller or larger. They can contain a different repertoire of proteins and mRNA. And this is why this is so exciting and open, and I'm so enthusiastic about this, because we don't really know how, in response to the same stress, muscle cells or cardiac cells or brain cells respond to the same stress. This is something that we know it happens, but we don't know the details of it at the mechanistic level. And knowing that, I think it would be really important to understand the basis of stress at the molecular level and figuring out maybe how to deal with it.
So you don't know if you had a previous heart attack or medical condition that would have made one part of your body.
That's another great question. In culture, if you stress cells repeatedly, they can restore to homeostasis after one stress, two stresses. If you repeat the stress, they can't anymore. So there is definitely like a time dependent, like a memory of the stress in some conditions, bless you, not in all of them. But that's something, again, that will be super interesting to find out. And we have more data in neurons about that in relation to neurodegeneration, and I'll show you some of that data in a minute.
What do you mean by continued stress, one part after another, or stress this week, a little bit next week, or
Both.
both?
Both.
And what happens when they can't absorb anymore? That's what I was afraid of.
The cells die if they can't handle anymore. And this is one of the mechanisms underlying neuronal death in neurodegeneration. Yeah. U2OS cells, they're a type of immune cell that's just, they're very round. They're just kind of a nice cell line to work in a
Those from fruit flies?
These are not from fruit flies. No. The same process occurs very similarly in fruit flies, in yeast, in worms, in fish. It's evolutionarily conserved, but the data I'm showing you is from human cells. Yeah.
You brought up sorbitol. So is there also like all your artificial preservatives or your artificial sweeteners?
We don't know the answer to that. We don't know the answer to that. It's like I said, it's an amazingly unexplored field. I would also say don't worry, because typically in the dish we manipulate with very high concentrations.
But higher than you would get in
higher than you would get in the chewing gum.
Let's stay away from chewing gum.
Well, I stay away from chewing gum for a number of reasons, but I stay away from sweeteners as well. I don't like anything fake, but that's my personal choice. We don't have real data to sway you away from that. So, just take it as a possibility that needs proper investigation.
So that was just being able to show us how you artificially create stress.
Yeah, how we model stress in a dish, and we can do that in animals as well. And I'll show you how that relates to neurodegeneration, which is the disease I've spent most of the last 15 years studying. All right, so in the past 15 years, it has become more and more evident that stress granules are part of our quintessential form of stress response that cells utilize to deal with an insult. But it's also become clear that if that stress is prolonged or repeated or doesn't go away, there is now a connection to disease. There is a connection between stress granules and cancer. There is a connection between stress granules and neurodegenerative disease. And this is the connection that I and in my lab, we've spent a number of years investigating. And data from other laboratories and my own show that, okay, now this is a little bit busy, so let me take you through. So this is a generic neuron with a nucleus. And a bunch of these extensions are called neurites. The shorter one are called dendrites, and they typically receive inputs from other neurons. And the longer one is called an axon, and that's the mechanism by which neurons talk to other neurons in the central nervous system. So what we know that under conditions of stress, if you look in this little inset over here, under stress, those stress granules that you looked at in the previous slides form, and then if the stress doesn't last for too long, you're going to see them there, kind of dynamic, trying to keep the cell from being overwhelmed. And if the stress goes away, they follow this route over here, where the stress granules disassemble and the proteins and the RNA go about their normal business. But if the stress is prolonged, or there is additional, there are mutations, I consider the aging process as a contributor to stress, because we know that certain proteins that deal with stress response are expressed at lower level. There is more oxidative stress. In those cases, these RNA stress granules do not dissipate anymore. They don't disassemble. Instead, they harden. And what is really interesting, and it's become apparent in the last also about 15 years, is that these RNA binding proteins that are part of these granules, like the proteins I showed you assembling in the previous slides, they have what are called prion-like domains, or sticky protein domains that make proteins prone to aggregation. So here you have a ball of proteins and RNAs, and they can't really dissipate because the stress is around. And then their intrinsic protein structure makes them sticky to each other and aggregate-like, and they form more hardy RNA protein complexes that do not go away. So they become large like this, and then they become modified and very hard to get rid of. And there is increasing evidence that some of those pathological aggregates in cells that you probably have heard about, like amyloid in Alzheimer's, Lewy bodies in Parkinson, other cytoplasmic aggregates in amyotrophic lateral sclerosis or frontotemporal dementia, originate from granules that haven't dissipated because the stress was around, and they hardened into proteinaceous aggregates. There is quite a bit of evidence for that. There is also some controversy, but now a lot of our interest in the field has focused on this process. How is it that these stress granules that initially formed to be protective are hardening and could become damaging and play a role in the pathology of neurodegeneration? So indeed, in 2006, so only 15 years ago, this seminal paper was published from a group at University of Pennsylvania. And it shows in panel G here on the left, this is a brain sample from an individual that died of old age, did not have frontotemporal dementia. The two samples on the right come from brains of patients that passed away from frontotemporal dementia. And I hope you can appreciate that this dark stain, which is supposed to be in the nucleus, in the patient sample, comes out of the nucleus and forms these weirdly shaped aggregates. This is a hallmark of neurodegeneration. Protein aggregation is a hallmark of neurodegeneration, can be seen in the brains of patients that pass away from neurodegenerative disorder, whether it's Alzheimer's, Parkinson, dementia, or amyotrophic lateral sclerosis, or others. And what this seminal paper showed, this was a review published in 2009, but this discovery was made in 2006. What was discovered is that a major component of these aggregates that are found in patient brains contain an RNA binding protein called TDP43. I have to tell you, human protein names are not very inspiring or fun. I'll tell you, fly protein names are much more fun. But this is an RNA binding protein that can associate with those granules that I showed you earlier. It's not critical for forming them, but it's regulating them, and it associates with those granules. And here it is in these pathological aggregates in brains of patients that passed away from FTD.
So that
Yeah.
those two cells on the right are not functional? that we have them?
They are dead. They are on their way to die, and the tissue comes from post-mortem brain samples of patients that died of frontotemporal dementia. So if it, in vivo, in the human brain, it's hard to know when a cell is still alive, functioning correctly, or dead. We don't quite have the tools. This is why we have animal models, to go in animal models and recapitulate the molecular events that occur in a human neuron. So then we can understand, when do cells become dysfunctional? When do they die? At what point in the stress response? But what is so remarkable about this protein called TDP-43 that I dedicated the last 15 years of my life is that not only that, it's mutated and can cause ALS, it can also cause Parkinson. But even if it's not mutated, the wild-type TDP-43 protein goes to those aggregates. And if you look here on the bottom of my slide, 97% of ALS patients, if you take out their spinal cord post-mortem, you section it and you look for protein aggregates, 97% of those spinal cords will have wild-type, not mutant TDP-43 aggregates. This is unheard of, to have a wild-type protein basically sucked into these protein aggregates that correlate with pathology and neuronal death. So that told us in the field that this is a very special protein. It can sense what is stressing the cell, even if it's not mutated itself. And it knows to go to these granules, and then it ends up in these pathological aggregates. So I thought, if we start making models and studying TDP-43, we can understand what goes on in 97% of ALS patients. Even more remarkably, 45% of frontotemporal dementia patients have protein aggregates that contain wild-type TDP-43 protein. And to make it even more interesting, in recent years, up to 50% of Alzheimer's patients, they may have plaques, but they also have wild-type TDP-43 containing aggregates. Parkinson patients have TDP-43 containing aggregates. Even football players that keep injured and develop chronic traumatic encephalopathy have wild-type TDP-43 containing aggregates. So I love this protein. It's super special. And I'm hoping that if we and others can figure out how it works under normal conditions and what happens to it under stressful conditions, we can figure out ways to stop and potentially reverse the degeneration process. So how do we study this? So I told you that when I studied my postdoctoral training, there were a few Drosophila geneticists that, typically Drosophila has been amazing for studying basic biology, development, but people started developing models of human disease. And I know that you and I and a fly don't look the same, okay? But we're actually much more similar than we think. I like to tell my students that, flies are little people, and of course it's an exaggeration, but let me tell you some interesting facts that you may or may not know about. There were 6 Nobel Prizes in physiology or medicine awarded to Drosophila geneticists for their seminal discovery of fundamental biological processes. It started in early 20th century with Morgan and Muller, who are sort of the luminaries in the Drosophila genetics field. In 1995, when I was a graduate student, 3 Drosophila geneticists received the Nobel Prize in Medicine for their discovery of genes in Drosophila that are conserved in humans. So as I mentioned, although you and I and flies don't look the same, the way we develop from a single fertilized embryo to how we look today with an anterior and posterior and a dorsal and a ventral side, the genes that control this development were discovered in flies by these 3 geneticists, and they are conserved in humans. So the way we come about to be who we are developmentally utilize the same gene cassettes that flies do. And we learned about those genes from flies. So next time you want to swat one, and you'll see how hard it is, because they're actually smarter than we think, please think that a lot of what we know about ourselves and human biology comes from those tiny, annoying little flies. There are other discoveries that are worth mentioning, but I just want to focus that in 2017, and that's recent enough that maybe you remember, these three Drosophila geneticists received the Nobel Prize for discovering the molecular clock. So, we sleep and we sleep at night and are active during the night, hopefully. The gene expression patterns are responsive to day and night. The circadian clock is critically important for our livelihood, for our health. There's evidence that even some medical treatments should be given at a certain time of the day because the circadian clock is important. The molecular components of the circadian clock were discovered in the fruit fly. They're called period and timeless, the genes that control the circadian clock. So it wasn't discovered in mice or in humans, it was discovered in fruit flies. Okay? So these are just some examples of seminal contributions that fruit flies made to our understanding of human biology. Let me tell you a little bit more. I like to tell people that in my lab we take a fly to human approach because there is a high degree of gene conservation. 70% of human disease genes have an ortholog in the fly. At the genetic level, we are incredibly similar. And also there are similar, although no identical systems. Flies don't have a liver. They have something that looks like a liver and functions like a liver, but doesn't look like a liver. But in terms of the nervous system, which is my main focus, there is a peripheral nervous system and a central nervous system that is organized in a manner that's rather similar to the way the nervous system is organized in humans and invertebrates. We have powerful genetic tools. I can't go about and give you a gene therapy injection, but I can do that to the fly and really study what gene loss of function or gain of function can do to a neuron or to a cell. We can really go in mechanistically and figure out what genes do during the lifespan in different cell types. They also have a short generation time. In about 10 days, you can get a new generation, so you can study lots and lots of flies. You can grow them in a lot of containers. And I really meant to bring you some flies, but I forgot yesterday in a hurry. And they don't cost as much as a mouse. I have probably 1,000 fly stocks in my lab, and it costs around $2,000 to $2,500 to just maintain all of them. But it's way less than you would pay for maintaining mouse models. So it's relatively cheap and it's much, much faster. So how did we model ALS 15 years ago in the fly? We did something that, if I go to Whole Foods, I love Whole Foods, but if I go to Whole Foods and say I make genetic modified organisms for a living, they'll probably kick me out. That's what we do, right? We make genetically modified Drosophila, and we do what we call we humanize them. We put human TDP-43 in fruit flies, in their motor neurons, or in whatever cell we want. We have the genetic tool sophistication, ability to manipulate individual cells in a temporal manner, very specifically with human genes. So we modeled ALS by trying to recapitulate those aggregates that you saw in patient samples, overexpressing wild type or mutant TDP-43 in motor neurons. And what you're looking at here is on the left is a normal, this is what we call the thoracic ganglia of the fly. It's the equivalent of the spinal cord. Every little white dot you see here is the nucleus of a motor neuron. This is how the motor neurons are organized in a normal fly. On the right is the spinal cord of a fly where we're overexpressing TDP-43 in motor neurons. And what's the outcome in ALS? Motor neurons are dying. What's the outcome in the fly? There are fewer of those dots because there are fewer motor neurons because many of them have died already. So we can recapitulate motor neuron death. We can also look at individual synapses, the neuromuscular junction synapse. As motor neurons touch muscles, we can look at various molecules that are in the motor neuron or in the muscle, and we see various morphological and functional abnormalities. We also see a reduction in lifespan. If you treat a fly nicely in the lab, it can live up to 80 days, and then a few of them make it to 100. If you overexpress wild type or mutant TDP43 in motor neurons only to mimic what happens in patients, you can see a significant reduction in lifespan, just as it happens to patients with ALS, which, by the way, survive on average 2.5 years after diagnosis. But if we're talking about modeling ALS, which is a disease that leads to paralysis and death and affects locomotor function, we wanted to see whether our little flies show locomotor defects. So I'm going to start these two movies. And what you're going to be looking at is our larvae on their way to becoming adults that express human TDP43 just in their motor neurons. The rest of the animal is normal. And the animal on the left is just the disease model, just expressing human TDP43. The animal on the right expresses human TDP43, but we administered a drug that we found it to be what we call protective. I hope you're going to see as you look at them that the one on the right is able to move better. So let me start the movies. Maybe I can do this. Oh, didn't do that. Let me go back here. Oh, I see. So I'm going to start them at the same time. So what you're going to see is that we flip the larvae. Sorry, I'm having trouble with the mouse. I'm not a PC person. Okay, so this is the one with the disease, and this is the one with the disease that ate a good drug. So as you compare the left and the right, I hope you can see that one on the right already turned back and it's moving, and the one on the left, it's still kind of struggling and dragging his posterior end around. So were you able to see that, or should I play it again? Should I play it again?
Yeah.
Okay. All right. Once this is okay, so I'm going to start. This is the disease model, and this is the disease animal that ate a protective drug. So we're turning them back. It's just like we're giving them a challenge. And take a look, the one on the right has turned around and it's moving, while the one on the left is still struggling. So it's visual, it's qualitative, but we can actually quantify this behavior by measuring the time it takes for the animal from the flipping to the time it turns back and starts moving correctly. Yeah.
What is the drug that you use?
In this particular case, it's an anti-diabetic drug. Anti-diabetic drug.
Okay.
We've published that work, and I'm happy to tell you more about that. So this is one of the assays that we use to seek drugs, either FDA approved or brand new small molecules that have never been pushed to the market, but could have a protective effect in our model. So this is the model that we generated, and I hope you can see that the phenotypes, as we call them, or the consequences of expressing human TDP43 in motor neurons cause consequences that are similar to what happened to ALS patients. Motor neuron death, locomotor dysfunction, synaptic dysfunction, reduced lifespan. So what do we do with this model? We do a lot of fun stuff. We're very interested in molecular mechanisms. I'm not going to bore you with RNA processing, but I'm going to show you some images of how we study mechanism. We're doing all kinds of genetic screens to identify what genes may change this phenotype, make it better or worse. We're doing drug screens using that assay I showed you. And we have some FDA-approved compounds that look promising, and we also have brand new small molecules that a colleague of mine in chemistry has synthesized, and we just got a DoD grant to pursue those as potential therapeutic leads. We're also interested in neuronal metabolism. Neuronal metabolism has been understudied, but we and others have found a number of changes in neuronal metabolism that we think could be harnessed and try to slow the disease progress and potentially restore neuronal health. So I want to take you back to this idea of ribostasis or homeostasis at the RNA level, which I brought to you in the beginning, that when cells are under stress, RNA stress granules form. I just have a different cartoon here that incorporates TDP43 now. So upon stress, stress granules form. My favorite protein, TDP43, is present in these granules, and they initially form as a protective response. However, if the stress doesn't go away or additional stresses are occurring, these RNA stress granules harden and they can sequester proteins and RNA and take them away from their normal function for much longer than they should. So one of the obvious consequences at the molecular level is that the RNAs that are being trapped in these hardened granules are not being made into protein like they normally should be. So the ribosomes are the cellular structures, is the cellular machinery that makes RNA into protein. So our hypothesis is that as these granules harden and they sequester RNAs, these RNAs will not be present on ribosomes. So they will be less available to ribosomes for making protein. And that's as far as science I'm gonna go, and I'm gonna show you some fun diagrams. This is how we look at big picture, what happens in terms of big picture gene expression changes in motor neurons. So what we can do in the tiny fly, we're just manipulating motor neurons. This is what's so cool. So then we pull the ribosomes just from motor neurons with a special technique. And then we look at the RNAs that associate with the ribosomes in a normal motor neuron. And we find that those RNAs encode things that have to do with mitochondria, metabolism, energy production. Makes sense. Neurons are highly energy demanding. Proteostasis is just another way of saying protein homeostasis. So typically neurons are occupied with RNAs that are important for energy and protein folding and just overall homeostasis. When we do the same experiment and we pull down ribosomes from animals that express wild-type TDP-43 or mutant TDP-43, we see huge changes in gene expression. RNAs are being sequestered and not properly made into proteins, and those go into different pathways that have to do with RNA metabolism, neuronal metabolism, mitochondrial metabolism. So this is the kind of big picture or the big data approach that allows us to get a view from the top of what's going on in these neurons. And then we can go individually, gene by gene and pathway by pathway, and validate, what can we now tweak to make this neuron healthy again, to make these flies live longer again. So this is one of the approaches that we take in my lab. But then I told you, we're not just studying fly biology for fun. We are interested in validating these in human patients. Are these meaningful in any way to the human disease? So we do all this molecular and genetic work, and then we look at that little larva and we dissect it out. And these little lines are motor neuron axons, and these little beads here are the neuromuscular junctions that the motor neurons form on muscles. And then we validate our findings. We look at what's altered in the fly, and then is it altered in patient tissues as well? So here's how we do this. And here are some examples of what we published. If you just look here on the bottom, here's the fly spinal cord. The red staining is for a protein that we found to be altered in the way I showed you earlier in the fly. So then we wanted to see, you see how there's more of it in these little donuts here? Those are motor neurons in the fly. I hope you can see there's more protein in the disease compared to the left. The label here is missing. To the left, it's a control. To the right is an ALS fly. I hope you can see there's more of this protein here. So now let's look at spinal cords. From healthy controls to the left and ALS patients to the right. This is the human homologue of the fly protein. And I hope you can see that it's also increased in motor neurons as it is in the fly. So this is our first validation that the molecules that we find altered in the fly model are also altered in a similar way in the patient spinal cord. Here's another example. And this, I just have a cartoon here. This is a neuron, and this is its synaptic terminal. We found in the fly that the specific protein that controls synaptic vesicle turnover or synaptic transmission is reduced in the fly, and we figured out the mechanism. The same happened in patient-derived motor neurons. So this was another example, and we have other published examples where again and again, what we find in the simple fly using these mechanistic big data approaches validate in patient tissues, whether the spinal cords or brain tissues or neurons derived through stem cell technology directly from patients. So this gives us hope that we can figure out mechanistically what happens in the fly, and then we can go to human tissues and hopefully validate. And so far, it's been amazing. And we hope that these findings are giving us new clues as to how we can intervene to save these motor neurons from dying. And we're also developing models of frontotemporal dementia, but I don't have time to show you. So really quickly, here's the data from our aging grant. So last year, we applied for what we call a pilot grant or a seed grant from the Innovation in Healthy Aging Initiatives at University of Arizona to study the formation of granules. We know they're involved in disease through the perspective of TDP-43. But we don't know much about, we know that aging is a big risk factor for neurodegeneration, but we know almost nothing about the dynamics of stress granules in aging. So we thought we're going to use the mighty fly to study stress granules in aging, under normal aging conditions, under stress, and then if we see some differences, we can then figure out what's in there, how do these stress granules change, and potentially improve the aging process and make it healthier. So first, I want to show you some very preliminary data, unpublished data, and it's not yet quantified, but I think visually it'll give you a pretty good idea. Here is a portion of a brain of a young fly, 1 day old. And in this case, I have magenta tagging a protein that goes into those stress granules. I showed it to you before in red or green, now it's in magenta. And basically, it looks like a bunch of donuts. Those donuts are all kinds of cells in the fly brain, neurons, glia, just like in the human brain. And the donut hole is the nucleus. So you kind of see the protein in the cytoplasm. But here, a 60-day-old brain, relatively old age, probably over 80s corresponding in a human. And I hope you can see that the distribution is different. And I think I'm hoping you can see that we have this puncta forming here, kind of resembling those stress granules that we saw in cells that were forming under oxidative stress. So there's no stress here, except that we dissected the brain out. That probably was a little stressful to the fly. But I hope you can see a change in localization, that we now see some puncta forming over there. How about under stress? We now looked at the middle age. This is a normal fly, and it comes reasonably okay. You see the cells, and now at 30 days, you can see some punctate staining, and there is a specific region in the brain, which I don't know yet what it is, that seems to show some special staining. We stress these flies, not the same fly, of course. We have normal flies, we dissect them out, we look at the granules, and then in this case, the stress we applied was heat shock for 1 hour. And after 1 hour of heat shock, we took the flies out, we dissected the brains, and we looked at the granules. And I hope you can see there's a lot more of this and a lot more puncta. So the fly brain of a middle-aged fly responds in this way to heat shock. We're doing a bunch of other stresses. And then we looked, this is a normal fly. In flies, we have a mutant called Methuselah, for good reasons, that is long-lived. This mutant fly lives 30% longer than a normal fly. And we took a middle-aged Methuselah mutant fly and we looked at granules. First of all, I want to point your attention to the no stress versus stressed. There is a bit of a response here, but I hope you remember it's not as dramatic. So I put them all together here. So normal flies to the top, long-lived or Methuselah flies at the bottom, no stress on the left, and stress on the right. And although this is preliminary and we haven't quantified it, I hope you can appreciate that the normal fly responds really dramatically to stress. But a fly that is resilient to stress and is long-lived by 30% and is also healthy living in this fly, it doesn't mount as profound of a stress response. So this is just a glimpse into what might stress response look like in an animal that is long-lived. Now, I don't know what's in those granules. These are kind of the next steps we're going to do. I don't know if this is just a heat shock response or there may be other stresses that we can do that. But this is where this project is going. So right now, we're going to be interested in figuring out how is the stress response difference in a normal animal versus a long-lived animal. How is the stress response different during lifespan, if it is? Identify the molecular components of the stress response, and then go back and manipulate them genetically and see if we can manipulate the stress response to make a normal fly a long-lived fly. That would be the ultimate goal. And we have the genetic and molecular tools to do that. So I just want to summarize. I hope I got you excited about stress granules. Just like their name says, they form when the cell suffers an insult, and their main job is to regulate what we call cellular homeostasis. They assemble upon stress, and they disassemble when stress goes away. However, if the stress is prolonged, or doesn't go away, or is repeated, there is evidence that these stress granules mature into hardy proteinaceous aggregates that correspond to some of those pathological structures we see in postmortem samples from neurodegeneration patients. We know the pathological aggregates impair gene expression, and we use those approaches in the fly to identify cell type specific changes caused by those, and then we validate those in humans. We also know that during aging, these stress granules exhibit an age-dependent dynamics, and we're going to go after the molecules that are changing during aging and under different stresses. And we're very interested in this long-lived mutant, Methuselah. There are others. There are also dietary interventions that can cause a fly to live longer. So we're going to look at stress granules in that as well and see how they might differ. So if we could recapitulate the stress response from a long-lived fly into a normal fly, we hope we can change the trajectory in terms of lifespan and health span for those flies. And these are some of the future direction, molecular investigations. And I just want to thank, these are some of the students currently in my lab. The work that I showed you on stress granules is done by Eva Orozco, who didn't send me a picture in time, and Mi-Yun Tran, shown over here, who's graduating with honors, and is also a Roby medal winner this year and outstanding senior in my department. And various other students and technicians in the lab, and also thanks to many funding agencies that are supporting our work, and a pilot grant from Innovations in Healthy Aging that I hope is just the beginning of a really fun journey into stress resilience at the molecular level. And with that, I want to thank you, and I'm happy to take more questions.
So Maya will ask the online questions first.
Oh, okay.
And if you could just repeat
Okay.
what they are.
All right.
Thank you. So I do have a question that came in asking about the oxidative stress that you created in your videos. When would you see that kind of oxidative stress occur naturally in our life?
During aging. The cellular pathways that deal with oxidative stress are altered at the level of gene expression as we age. They're less powerful. They're less powerful at dealing with oxidative stress. Injury can cause oxidative stress. Infection can cause oxidative stress. A bad meal can give you a tummy upset and cause oxidative stress is very common. The question is how high, how long can the cell deal with it? So the normal process of aging is to produce less of those molecules that deal with stress. And then in addition, as I mentioned, disease or injury.
Great. And then my other question that came in was just about the impact of the lifespan of a fruit fly. Does that impact the amount of research just due to the shortened lifespan of the fruit fly? Or I know you've sort of, it seems like it's sped up, right, for their lifespan. Does that make any difference in what you're seeing with humans?
Let me see if I understand this correctly. Maybe you can say it again, but
Sure.
as I understand it, is the comparison between the long-lived fly and the normal fly and the relevance to humans. Is that the question?
Well, yes, on the impact of your research with the stress granules.
We don't know yet. I hope it's going to be great. I mean, first of all, we know almost nothing about it. And we do know that this dietary restriction that people are adopting in their own lives have been validated and discovered in simple models like fruit flies and C. elegans. So whether there is a direct parallel between what we find in the flies and the humans, we'll never know until we try it. But so far, it's been an incredible validation between the findings from the fly to the human. And this is not to, I'm sure you're all familiar with mouse models. This is the most used animal model in research, rodents. And this, I'm not here to bash the rodent models, but I teach a class and I tell my students it's a great time to be a mouse because we can cure every disease in the mouse. However, we haven't been very good at translating those findings from the mouse to humans. In ALS, all clinical trials coming from mice to humans failed miserably one after another. We don't know why. Doesn't mean the mouse is a bad model, but I think it's time to take a bit of a out of the box approach and study other models that we already have evidence that they have contributed to understanding of human biology and human disease and study those as well. So I can't make you any promises, but I think it looks pretty cool.
Great, thank you. Thank you so much for being here today, Dr. Zarnescu. I want to thank you from our online audience, and I'll let you go ahead and take any extra questions.
Of course, thank you. I think you had a question? Yeah.
Yeah, you mentioned changing the diet in fruit flies. What else does a fruit fly eat other than?
It eats protein, fat, and carbs just like we do. Yes.
Well, how does a fruit fly get protein out of like a banana or?
Well, in the lab, we treat them like in a gourmet restaurant. Okay,
Like,
so we give them really nice food that contains yeast extract, cornmeal, molasses, and a little bit of agar to solidify. So imagine like a really gourmet polenta. That's what they get in the lab. So they get their protein from yeast, and we can change the amounts of protein, carbs, and lipids we can add. And actually, a few years ago, we published a paper in which we showed that a high carbohydrate diet in our ALS model can improve lifespan and locomotor function. If you give a high carbohydrate diet to a normal fly, that's not very good. That fly will get diabetes and will live a shorter life. But if you do that to a diseased animal, that will be protected. That will to the ALS fly. If you give a high carbohydrate diet to the ALS fly, the fly will perform better in locomotor assay and will live longer. And we did that with diet and also genetically. We manipulated glycolysis genetically, and we showed the same thing. And there is a very limited clinical trial in patients that shows that a high carbohydrate diet has a good outcome on patients. And let me tell you, after we published our work, I received a lot of phone calls and people telling me, I eat a lot of pasta and I'm doing really well. I'm like, I'm not a physician. We need to do a clear clinical trial. But I got to tell you, we can find out a lot of interesting things just by manipulating the simple fly.
I want to thank everyone for being here today to see Dr. Zarnescu. We are going to be back on tomorrow morning for Take Charge of Your Healthcare. So please join us. That is for Friday, April 15th at 10 a.m. And we'll see everyone later.